Hi, thank you so much for being here. My name is Dion Rossiter. I am the Executive Director of Science at Cal. We welcome you again to one of our many Midday Science Cafes where we team up with both UC Berkeley and Berkeley Lab scientists. Today we have synthetic biology for a sustainable future, and we have two very talented and brilliant scientists here to joining us today. I'm going to go ahead and give a quick land acknowledgement. UC Berkeley and Berkeley Lab recognize that Berkeley sits on the Huchun territory, the ancestral and unceded land of the Ohlone people, the successors of the historic and sovereign Verona Band of Alameda County. This land was and continues to be of great importance to the Ohlone tribe and other familial descendants of the Verona Band. Every member of the Berkeley community has and continues to benefit from the use and the occupation of this land since the institution's founding in 1868. By offering this land acknowledgement, the Berkeley community not only recognizes the history of the land on which we stand, but also recognizes that the Ohlone people are alive and flourishing members of the Berkeley and broader Bay Area communities today. Thank you so much for allowing me to make that acknowledgement. Just a quick little preview for what we have in store for the rest of the summer. We have our July and our August Midday Science Cafes, one on negative emission technologies and one introducing some of the greatness and the wonders of the periodic table. So please save the dates, write those in your calendars. Science at Cal and Berkeley Lab has a series of additional events that are happening throughout the summer. So visit our websites, which you'll see in just a minute, to learn more about that. You can find, Science at Cal celebrates science through public programming, science cafes, lectures, festivals, and more. You can visit us to learn more about everything that we do at scienceatcal@berkeley.edu. You can email us. You can find us on Facebook, Twitter, Instagram, whatever your heart's desires. We are hoping to be live and in person in the fall, so stay tuned for some of our upcoming programs, our lecture series, and our series featuring graduate students. We're hoping to get those all back either on campus or within the community. I want to remind everyone that we are taking Q&As at the end of every single presentation. So we really encourage you, if you've been to any of our events, you know this, we encourage you to ask your Q&As within the chat box or the Q&A box, and we'll get to those both after each one of the talks, and also at the end we'll have an extended Q&A. We'll be able to answer all of your questions as a group. Also, I want to remind everyone that this is being recorded. So you can go ahead, we'll share it with you at the end of our program, and it also will be posted on both of our websites for you to find. You can find all of our programs, actually, all of our lecture series, all of the Midday Cafe series on our website and on our YouTube channel. So right now I'm going to hand things over to Jocelyn, who's going to introduce Berkeley Lab, because again, this is a really, really great collaboration between both Berkeley and the lab itself. So let's get there. Go ahead, Jocelyn.
Thanks, Dee. As Dean mentioned, my name is Jocelyn Delgado, and I'm the Community Relations Administrator at Lawrence Berkeley National Laboratory. Berkeley Lab is one of 17 U.S. Department of Energy national laboratories across the country, and we're supported by DOE's Office of Science and are managed by the University of California. All of the research we conduct is unclassified. And since our founding in 1931 by a UC Berkeley physics professor named Ernest Orlando Lawrence, Berkeley Lab's been dedicated to advancing the scope of human knowledge and seeking science solutions to some of the greatest problems facing humankind. This year marks the lab's 90th birthday, and as we celebrate our past achievements and imagine what discoveries the next 90 years might bring, we hope you'll visit our 90th anniversary website, which is berkeleyLab next90.lbl.gov. which features many opportunities to engage with us, including a lecture next Friday, June 25th, about how researchers are using machine learning, a branch of artificial intelligence, to spur discovery and innovation in a diversity of fields, from energy and the environment to biology, astronomy, and beyond. Today, Berkeley Lab researchers develop sustainable energy and environmental solutions create useful new materials, advance the frontiers of computing, and probe the mysteries of life, matter, and the universe. Our main campus is nestled in the Berkeley Hills above the UC Berkeley campus. We employ approximately 4,000 people, about 1,700 of whom are scientists, engineers, and faculty members. More than 500 of our employees are undergraduate and graduate students, meaning these are scientists who are beginning their research journey. Berkeley Lab's proximity to Cal and our close ties to the UC system create a unique and synergistic environment for scientific discovery. A number of the lab's researchers are affiliated with one of the UC campuses as students, postdocs, and professors with joint appointments at the lab. And as you can imagine, Berkeley Lab's relationship with UC Berkeley is especially close, and our institutions have joined forces to advance science across many frontiers. One of the main motivations for creating our Midday Science Cafe series is to share with you examples of compelling and complementary scientific research from both of our institutions. We hope you enjoyed today's presentation on synthetic biology. Thanks so much, and Dee, back to you.
Wonderful, thank you. So at this time, I'm going to stop sharing my screen, and I am going to introduce Yuzhong Liu to join me and share her screen. So let's get the bio in front of me. Yuzhong Liu obtained her PhD in chemistry from the University of California, Berkeley, studying dynamic porous materials for guest capture. Interesting, you might learn what that means today. She's currently a postdoctoral fellow at the Joint Bioenergy Institute, or JBEI, at Berkeley Lab, focusing on the sustainable production of complex natural products through foreign pathways expressions in microbial hosts. Her work has been featured on Forbes, Science Daily, and on the cover of other leading scientific journals such as Science and the Journal of American Chemical Society. In the meantime, Yuzhong also serves as the Associate Director of Commercialization at JBEI to help transition technology from the research lab towards real-world applications. It is so wonderful to have you, Yuzhong. I cannot wait to hear your presentation. So go ahead and teach us something. Take it away.
Thank you, Dee, for the introduction. Hi, everyone. My name is Yuzhong. I'm a postdoctoral fellow in Professor Jay Keesing's lab at UC Berkeley and JBEI. And today, I would like to tell you a bit about how we can harness the power of synthetic biology towards a sustainable future. Now, before we get started, I want to pick your brains and ask you, which of these following items do you think are usually made from fossil fuels? These are, very much everyday items you would use. You have your answers. I don't know if this is a surprise, but yes, all of them are usually made from fossil fuels. And this is just a simple demonstration that our daily lives are very much heavily dependent on fossil fuels and products. And we all know that climate change is happening at an unprecedented pace, and we're in urgent need for clean and sustainable alternatives for both energy and product purposes. And that leads us to the mission of JBEI, where we develop not only the basic science, but also advanced technologies to convert biomass. So for example, plants, and in particular, polysaccharide, these complex sugars, as well as lignin, which is the woody and rigid part of the plants that are typically non-edible. We break them down to access the carbon and then feed that to genetically engineered microbes to produce biofuel and bioproducts. Now, this might sound like a very complex challenge, and it is, but that's why 5 different research divisions at JBEI work very closely together, where we start with feedstocks, where we have the plants, we deconstruct them to access the carbon source, and then we modify the microbes to produce biofuels and bioproducts. So this process is assisted by the technology development that we have, for example, the software, the automation platform, to ensure that our research can happen in an efficient and a high throughput fashion. Last but not least, we also conduct life cycle and techno-economic analyses to make sure that, oh my God, my cat, sorry. I'm so sorry. Can you see my screen now? Yes, okay. Yes, life cycle and techno-economic analyses to make sure that the products that we have are both economically and commercially viable and can be adopted into real world applications. So how does all of these work for synthetic biology? Now, shown here are two organisms, the yeast and E. coli. They are the most common host for engineering, and we can just call them bugs. And as you can see here, metabolism is very complex, even in very small microbes. But luckily, we can target specific pathways for different applications. So this is a zoomed in version of the pathway, the very complex pathway that we just saw. So for example, in this red box, we can look at the isoprenoids pathway and production. And these molecules are good for gasoline replacement and very interestingly for aroma and fragrance purposes. So yeah, indeed a very interesting class of molecules. And also we can engineer the pathway to make fatty acids, which can be used for various food purposes, for soap, detergent, and also cosmetics. So in addition to using entirely the native pathway of the bugs, we can also just hijack a part of the pathway and express foreign genes from other species, and then make the bugs make products that they wouldn't be naturally able to make. So for example, the polyketides here. So when all of these engineering and modifications are done, we grow the bugs in these bioreactors where a variety of parameters, for example, temperature, pH, oxygen levels can be very accurately controlled to ensure the maximum growth and productivity from these microbes. So the first example I want to show you is the production of biofuels. So we all know that global warming is very real, and it's largely due to the excessive carbon emission into the atmosphere. And we believe that biofuels can provide a viable solution towards carbon neutrality by fixating the CO2 from atmosphere into biomass via photosynthesis, and then convert the biomass into biofuels using the process that we were just talking about. And that can be the fuels for the various transportation schemes. And now the CO2 is being released into the atmosphere again, and then captured by biomass, thus closing the carbon cycle. And by now, actually, the biofuel production has been rather developed. And at the beginning, we were using sugar canes and other sugar-rich plants, for example, beets, and then ferment that sugar into bioethanol and biodiesel. And shown on the right is one of the largest biodiesel bioreactor in Brazil, where 75,000 tons of biodiesel are being produced every year. But naturally, you would ask, oh, why do we use all the sugars for fuel? But like, what are we going to eat? That's a very valid question. The food versus fuel crisis was raised at the beginning of this generation of biofuels. That's why now currently we're developing the second generation of biofuels where we use the lignin, the polysaccharide, the non-edible part of plants towards biofuel purposes. Now, this slide looks very complex, but don't worry, it's kind of a showing off slide just to show you this large variety of molecules that we can make biosynthetically for fuel replacement. So for example, the short chain alcohols on the left shown here can be used for gasoline replacement. And then on the other hand, these longer chained strained molecules have higher energy density and can be used for things like jet fuels. And indeed, farnesene, which is the saturated version of this molecule, was the first bio jet fuel that was commercialized. And currently, and looking into the future, we're also developing platforms where we can make designer fuels, where we design the structures towards specific fuel properties. And now I'm gonna switch gear a little bit and tell you about the second example here, about jeans, which a lot of you are probably wearing at the moment. So statistically, apparently, every American has 7 pairs of jeans, and around the world per year, approximately 6 billion pairs of jeans are being made. And that gives us 50,000 tons of indigo being used every year. It's probably not hard to guess. I'm going to say that indigo is made from fossil fuels as well, but you might not know is that for each equivalence of indigo, you need half equivalence of cyanide, which is very toxic, to make indigo. In addition to the hazardous chemicals being used in this process, the water pollution also is very harmful for the local environment. That's why there is a huge push in the Greenpeace movement to detox the denim industry. So this is also another example where synthetic biology can provide a solution and an environmentally friendly replacement for indigo, where we have this natural dye indigoidine. So in order to make that, we can utilize environmentally friendly manufacturing processes using non-portable water and the lignin biomass. We engineer the buck to be able to produce very high quantity of the indigoidine. And as you can see here with the bioreactor, it's a very scalable process. And at the end, we were able to purify this blue pigment and potentially as a drop-in dye to fit into the currently existing infrastructure in the dyeing industry. So this is still very much ongoing. But I'm just going to share with you one fun day that we had in lab where we thought, why don't we just give it a try and see what happens? So on the left, you can see we collected the cells where this indigoidine is being produced. So these are just all cell pellets. And then we collected that, and then you can see we dyed the sleeve of the t-shirt, and it is very much blue. So a lot of the dye property testing of this molecule is very much ongoing. But so far, it seems like all of this is still very far from our daily life. So I'm going to show you next what else we can make using synthetic biology, and it is Hoppy Beers. Yeah, it's very popular in the Bay Area, at least. So traditionally, you would brew the beer using yeast for the first step. And then in the second step, you would add the hops and then to make the hoppy flavor. So a couple of issues here is that depending on the batch of the hops, the hoppiness tends to be not very consistent. And then also the hop addition would use hop, obviously, and additional water. So researchers at JBay have also integrated the pathway from plants to make the hoppy flavors into yeast. So now we can not only combine these two fermentation steps, but we can also save the hops and the water to achieve a consistent level of hoppiness in these hoppy beers. And they actually founded a startup called Berkeley Yeast, and their beers are very much commercialized now. I know it's Thursday, but feel free to grab a beer at the end of the day. So that's the beers. And what else can we make? And we can actually make cannabinoids in yeast as well. So we all know that CBD is very effective and widely used for treating pain and anxiety and etc. But CBD and THC actually exist in very low quantities in the natural plant, cannabis. That's why the extraction is actually very problematic. But in order to assess its pharmaceutical value, you need, I guess, a very pure form of this molecule. That's why people have also turned to synthetic biology. But since these molecules are rather complex, the synthesis is very expensive. Now, with a very similar engineering concept, we copied the pathway from cannabis that makes CBD and THC and pasted that into the yeast host. And now using simple sugars and the bioreactor that we just talked about, now we can make THC and CBD in a very scalable fashion and potentially in an economically viable way as well. Now, researchers have also started the startup Dimitrex, and that is also localized in the Bay Area. So to wrap up, I know we heard a lot of fun stories today, but I hope I was able to convince you that synthetic biology does provide a viable solution towards a sustainable future, where we drive the technologies towards carbon neutrality and also provide sustainable alternatives to the currently fossil fuel-based products in a scalable and economical way. So yeah, with that, I would like to take any questions.
Thank you. Thank you so much. That was completely fascinating. We do have a few questions, and I will get to just a few before we hand things back over to Jocelyn. But first question is, what are the advantages of using engineered microbes over plants?
Yeah, that is a very good question. And it's super valid, especially when we look at the a couple of examples that we have shown today. With a plant that is easy to grow, that is cheap, and if the extraction process is very simple, yes, the plant would be the way to go. But often that's not the case, because plants are composed of many thousands of complex structures that are difficult to separate. So the extraction process typically is very laborious. So that adds to the cost of that. And also, when you think about the cultivation of plants, you think of it in a very vertical, horizontal way. So that would take a lot of the farmland that we could potentially use to grow crops for food. But then if you think about the bioreactors, as you can see in the picture here, we can very much expand vertically. So that wouldn't take as much space and in a more scalable way. And depending on the season, sometimes the harvest of plants are not very consistent. So that would lead to very much fluctuating prices in the market. And imagine if it's a pharmaceutical, you don't want your medicine to go up in price for the next year. And, so we're using the microbial production will be a very consistent and sustainable supply of these important molecules.
Got it. Great response. Very thorough. Thank you. So do you know approximately what percentage of clean energy mix for bioenergy is predicted to account for or desired to? Is there a percentage that we're shooting for?
I'm actually not 100% sure. That's a very good question.
Maybe Changman knows the answer. We can ask him later when he's up.
Yeah.
Okay. Another question. Are there breweries now that are using the new yeast in their hoppy brewer? There are. Yes. Which one?
So in Berkeley, the one that I have been to that they sell this is Fieldworks.
Awesome.
But they're available at many more locations. You can just go to Berkeley Yeast and they have a list of breweries that have their beers.
All right. So whoever asked that question, you've got your answer, Fieldworks, and also everyone else. It is Thursday, but Thursday, as we know, it's the Friday. So you guys go out and try one of these hoppy flavored beers. That's really cool. What about denim? Is Levi Strauss willing to use the blue dye for their denim?
That's a very good question. Actually, with this dye, we actually got to visit the headquarters of Levi's, which was super cool.
Ooh,
yeah. They're very excited about clean energy and clean replacement for dyes. And they're actually making a lot of efforts towards saving water and minimize the water pollution. So they would like us to test the dye properties first before we at the actual collaboration, but they're very interested in this.
Oh, love that. So the answer is yes, they are willing. Awesome.
Yes.
That's so great. Well, thank you so much. We're going to hand things over to Jocelyn and Changmin, and we will come back to answer more of the questions from the audience. And I just want to remind everyone, you know, you're seeing me in the chat, really encourage you to ask questions so we can get to these questions at the end when we wrap up. So thank you so much again, Yuzhong, you're wonderful. And we will hand things over to Jocelyn.
Thank you.
Thank you, Yuzhong, for that great presentation. And now it's my pleasure to invite our next speaker to the screen, Dr. Changmin Kim. Chaiman is a postdoctoral researcher at the Advanced Biofuels and Bioproducts Process Development Unit, aka ABPDU, which is located at Berkeley Lab, where his research spans a wide range of biological and chemical engineering. In particular, he has been focused on electro-fermentation, a novel technology that can overcome some of the limitations of conventional fermentation, leading to the sustainable production of high-value chemicals used in a variety of industries and products. Changman received a Bachelor's of Science degree in microbiology from Pusan National University, a master's degree in ocean ecology from Puhang University of Science and Technology, and a PhD in chemical engineering, also from Pusan National University in South Korea. Changman, over to you. Thank you for joining us.
Hello everyone, I am Changman Kim. Today I'd like to introduce an arising biotechnology. It's called electro-fermentation. We are targeting to improving the bioproduction productivity using the microbe electrode interactions. So white biotechnology or industrial biotechnology, it is technology are targeting to replace current fossil fuel driven industry. As you know, or as Yuzhong said, current fossil fuel industry produces not just gasoline or energy, but also the lots of the material for the current human life, such as cosmetic, plastic, drug, and clothes, stuff like that. However, this fossil fuel is limited resources and cause environmental problem. So industrial biotechnology try to replace this fossil fuel driven material to the bio-based or clean material. And most of the chemical conversion of the fossil fuel is usually multiple chemical reactions occurred in the chemical factory. This chemical conversion is already well established and also has a high productivity. But however, as you know, these can cause environmental pollution and also the concerns for the resource limitation. So Microbes cell factory, it's a biotechnology factory having the multiple chemical reaction inside of the microbe or bacteria. It is a relatively clean resources and we have unlimited material. However, it is significantly lower productivity compared to chemical reactions. So let's think about the energy dynamics for the microbial cell factory. Substrate is a starting material for the bioproduction. It is obtained by microbe, then the multiple chemical reaction would be occurred, and finally this substrate converted to the product. When we see the energy dynamics of the bioconversion, there are two types of the bioconversion. One is a catabolism. Catabolism is substrate has a higher energy value compared to the product, as much as this thermodynamic value. Thus, the remaining energy should be released out from the microbe during these catabolic reactions. On the other side, products have a higher energy value compared to the substrate, and then thus it is required to put more or additional energy into the microbe for the successful energy reaction. So it is very important to overcome this energy imbalance between the substrate and product or thermodynamic barrier. Yeah, conventional strategies to overcoming this thermodynamic barrier are mainly three methods in bioproduction. One is aeration, co-production, and chemical addition. Aeration is a powerful tool to provide the oxygen as an electron acceptor. However, it increases the aeration cost. Also, co-production is very helpful to balancing the energy state by producing low and high energy products simultaneously. But byproduct formation would be the problem. And chemical addition are attempted to make the energy balance inside of the bacteria. But however, it makes several issues like the cause the byproducts uncontrollable. So we need to find a new technology to overcome this overnight barrier. It has a cheap or controllable or no byproduct formation. In nature, there are only a few bacteria are able to transfer or accept the electron from the electrode. This electron transfer between the bacteria and electrode makes a chance to solve this energy difference problem by just transferring or providing the electrical energy transfer. There are two types of the electron transfer between the microbe and electrode. One is the direct electron transfer. It is mostly found in the Geobacter or Shionella species. Indirect electron transfer is identified in some bacteria species such as Klebsiella and Pseudomonas species. Electro-fermentation is a bioproduction strategy using this microbe and electrode interaction to conduct energy out or energy in during the catabolic or anabolic metabolism. So, or in this aspect, in the aspect of the microbe, this bacteria and microbe and electrode interaction could be the non-native microbial metabolism, but occurred outside of the cell membrane. So let's see the several case study about the applications of the electro-fermentation in bioproduction. So first case is a 2,3-butanediol production. 2,3-video is a platform chemical, can be used in cosmetic or tire polymer production. In this study, starting material was acetylene, having 22 reduction state. It is equal to the energy level. But final product 2,3-video has a 22 electron value. So in this study, additional electrons should be provided to overcome this energy difference between the acetylene and 2,3-video. And they run this electro-fermentation, and then they finally get the 2,3-video production in only with the electricity on mode. And next case study is 3HP and 1,3-video production. 3HP is a 3-hydroxypropionic acid. It's a platform chemical used in the acrylic acid synthesis or nitric glove synthesis. And 1,3-video is currently applied in PTT synthesis, which is used in the textile or car seat. Both chemical can be produced from the glycerol fermentation, but energy state is completely different with the glycerol. Glycerol has a 14 reduction value, but 3HP has only 12, and 1,3-video has 16. So to produce more 1,3-video, it is required to put more additional energy into the bacteria. Electron transferring improved the 1,3-video, as you can see the left side. So electricity on mode enhanced the 1,3-video production from 23 to 35. On the other side, 3HP is needed to remove the energy from the bacteria. The passive electrical mode works as an electron acceptor or energy acceptor, and then energy can be released out. Then 3HP production was significantly increased 12 to 21, as you can see the right side. As I previously said, there are only few bacteria has this electrochemical activity. So synthetic biologists try to insert this electron transfer pathway into the non-electrochemical microorganism, such as E. coli. After several studies, they are successfully inserted and activate this electron transfer module from the Shionella species into the E. coli species. And then there are several applications studied for the electro-fermentation using this synthetic electron transfer pathway E. coli. One of the case study is for the succinate production, using this synthetic electron transfer pathway E. coli. Actually, succinate is used in the food and drug industry. So this study's starting material is a glucose, which has a 24 reduction state, but succinate has a 28. 2-succinate has a 28 reduction state. Thus, this study they try to put the additional energy into the bacteria and then presented significant increase of the succinate production from the 18 to 30. Yeah, to conclude this presentation, yeah, current biotechnology still is not sufficiently powerful to compete with the fossil fuel industry. But however, the synthetic biology improved and engineered the microbe itself, like the super soldier serum for Captain America, and electro-fermentation also can give the electrical energy to overcome the current limitation represented as a thermodynamic barrier to improving the productivity, like a thunder hammer, like Murley. I wish this collaboration can beat the Thanos in near future. And thank you for listening.
Thank you, Changman. That was a great presentation. Those case studies really brought it home. We'd like to ask you a couple questions. The first of which is, what is the current stage for this electrofermentation technology? And are there any real applications?
Yeah, so that's good questions. So actually the concept of electrofermentation was just suggested only 10 years before. And now researchers are just demonstrate this concept works in the various bioproduction. And as I know, there is one company, it has a LanzaTech. They are trying to reduce the CO2 to synthesize volatile fatty acids by providing the electricity. Yeah, that's the only one is real application of this electrofermentation. But yeah, most of those bioproduction researchers are very interested about this technique because we can suggest we can provide the strategy for overcoming this thermodynamic barrier.
And what would you say is the biggest challenge of electrofermentation to apply real bioproduction?
Yeah, that is another good question because I already said in previous slide, there are only few bacteria does have this electrochemical activity. So that's why people are trying to insert this synthetic electron transfer pathway to the non-electrochemical active bacteria. So that could be the very, very limitation in currently.
Okay, thank you so much. And now I'd like to invite everyone to join us back on screen to kick off our Q&A session. Thank you so much, Changman.
Yep.
Hi everyone. So Changmin, could you answer that question that we asked earlier about what percentage of clean energy mix bioenergy is predicted to or desired to account for? Do you know?
I don't have a very good numbers of this answer, but still the fossil fuel derived industry, they have a very good productivity. So we are trying to increase our productivity to beat their industry, but yeah, still we have a very lower. So, but yeah, so in some case, sometimes they are overcoming this industry. The fossil fuel industry is using the biotechnology, but yeah, still we have most of the cases we are using currently, but yeah, so maybe synthetic biology or electrofermentation can be suggested for the more good technology.
Right. Great. And this is a question I think for all of us. Somebody wants to look up this topic later. Would you call what would you Google to look up more about this information? Would you call it synthetic biofuels? Is that what you would call it?
Synthetic biology, bioproduction, metabolic engineering. Yeah,
those are great. Okay, that's a good question for all of us. Like, if we want to learn more about these topics, it's a little bit. There's so much technical language in here, but I think that's great. Thank you. All right, so what would be the estimated cost for these products? how do they compare to crude oil? I don't know if we went over that directly. And I think that this could be answered by both of you, right?
Yeah, like Chairman mentioned earlier, so these products are still at a slightly, okay, slightly is a very strong word, but at a higher cost than fossil fuels. That's something that we work very hard to bring down the cost by increasing the productivity, the tighter. So the more we can produce, let's say in 1 liter of fermentation, the less the cost it would be. That's why a lot of the efforts are being made towards metabolic engineering and synthetic biology to try to increase the productivity of these microbes, and therefore we can bring down the cost in the very near future.
Thank you, Yuzhong. Our next question from our audience is, are there any bacteria that consume CO2 as a substrate to produce useful chemicals? That could be a question for either of you.
Yeah, that's true. So I said there is a LanzaTech try to synthesize such kind of the volatile fatty acid from the CO2, but the CO2 has a very low energy value, so we need to think about how we can provide the energy to synthesize a more higher energy value chemical. So yeah, that is a very key issue. So I think LanzaTech is trying to develop the process to synthesize the fatty acid from the CO2.
Okay.
I have an answer too.
Oh.
So there are these bacteria called cyanobacteria. They can natively perform photosynthesis. So imagine them as tiny little trees that are floating in the water. So they can directly take up CO2 and work like trees to capture them and convert them into other valuable products. And indeed, that is a very rapidly growing area of science where people try to engineer these cyanobacteria to make biofuels and other value-added products.
And how many species have electrical activity, would you say?
Yeah, I can say I cannot count these bacteria species exactly, but yeah, I in my experience, so there's only 2 or 3 species does have a direct electron transfer, but yeah, so some researchers are trying to develop the indirect electron transfer using the chemical, but still it has a very, very low. So that's why people are trying to insert a new or synthetic electron transfer pathway from the Schwannella species into the non-electrochemical bacteria.
Great, thank you. So we have a question that maybe you can better understand, Yuzhong. It says, do you think that yeast can hold big pathways? Does that make sense to you? It does. Okay, yes. Can you explain that to us and then answer?
Yes, yeast actually has a huge genome. And that gives us a lot of opportunities to play with the native pathways and also to plug in foreign heterologous pathways to make things that are unnatural to yeast. And one of the projects that I have in lab at the moment is to plug in more than 35 genes into yeast. It's a huge pathway and it's actually working pretty well. So to answer your question, yes.
Excellent, thank you. This question is for both of you. What can synthetic biology teach us about basic biology? What do you guys think? This is kind of a meta question, right?
Okay, maybe Yuzhong, you can first.
Okay. Yeah, so in order to understand, so we saw the metabolic map of the microorganisms. They're extremely complex, and a lot of these parts we actually don't know yet. So what we can do is we can work and engineer a part of this pathway and see what the effects, the consequences that tiny little engineering strategy can cost, and that will definitely help us understand how intricate and entangled these pathways work in microbes, to provide a much more general understanding on the global, I guess, an idea on the metabolism.
Yeah, my answer is yes. It's a very good answer, and the synthetic biology is the words of the synthetic biology itself is just putting in the non-native mechanism, non-native reaction into some kind of the bacteria or into the organism, microorganism. So yeah, so the synthetic biologists try to insert a non-native pathway into the non-native pathway or some another foreign pathway into the bacteria. And also I suggest this electrochemical interaction between the bacteria and electrode. This is also a kind of the reaction or mechanism for the bacteria. So we can call this is a synthetic biology.
Wonderful, thank you. I'll ask one more question. So is the indigo dye from the synthetic biology process used in the dyeing process in the same way as the plant-based indigo? And the person asking this question is asking because there's the dyeing process from the plant-based indigo requires a chemical reduction solution, which is still really polluting. So does that eliminate that polluting factor? Do you know?
Yeah, that's a very good question. And when we were trying to go around and introduce this dye to a lot of people, that's a question that always comes up. Yes, so at first glance, the plant seems like a very good alternative to the fossil fuel-based indigo. But at the same time, the extraction is actually very complicated. The content of indigo in the plant is rather low. So you will be cultivating this huge farmland of plants, but to collect a very small amount of indigo. And another thing is indigo is, if you look at the molecular structure, it's very rich in nitrogen. So you would need to feed the plant additional nitrogen source, and obviously a lot of water for these plants to grow well, and then taking up a lot of the land use that could be potentially used for other things. Now, for the indigoidine, I didn't really show the structure, but it looks, actually, it's a very different molecule from indigo, although the color looks very similar. It has additional nitrogens in this molecule, so that we think it might bind much better to the fabrics through hydrogen bonding, because there are more nitrogen atoms. And hopefully we don't have to go through the same dyeing process that indigo has to utilize, thus eliminating the water pollution there. But yeah, that's still ongoing, and we will test that.
Very interesting. Thank you.
And synthetic biology has been gaining a lot of new interest. What is one of the areas or fields that you're most interested in seeing the application of synthetic biology grow? This is a question for both of you.
Sorry, could you repeat the question one more time? My internet just...
Oh,
sorry.
What is one of the areas or fields that you're most interested in seeing the application of synthetic biology grow?
Okay, yeah, it's a very hard to answer the question, but yeah, we are interested. We are working in the white biotechnology to replace the current fossil fuel industry. But yes, synthetic biology is not just limited in the white biotechnology, but also it is spread on a lot of biotechnology, such as the medical biotechnology or green biotechnology, which are the plant biotechnology or such like that. So yes, synthetic biotechnology is just a tool, but it expands our biotechnology world to another industry.
Okay.
Yeah, I agree. And for the examples that I have shown today, I do believe that synthetic biology has a very wide application across many different fields, varying from the very expensive pharmaceutical products to things that are very relevant to our daily lives, like beers. So I do believe also the diversity of the applications is very important for the development of the field. So I don't have a favorite field, but I would say it's very relevant for a lot of fields.
And what inspired you to study synthetic biology? Again, question for the both of you.
That's a very good question, actually. So as Dee mentioned in my introduction, I was actually trained as a chemist for my PhD. A lot of chemists are interested in the total synthesis. I don't know if it's a concept that it's familiar to everyone, but basically you would go through tens, maybe 100 steps of chemical reactions to achieve a final synthesis of one molecule, typically for pharmaceuticals, or just a very interesting molecule that you wake up and you're like, oh, why don't I make this molecule today? But that process involves a lot of labor, a lot of hazardous chemicals, which the microbes can do in such an elegant way. You just plug in a couple of genes, and at the end, you already have the molecule. So to me, this is very interesting, very efficient, so that I don't have to do all the chemical synthesis. So yeah, it's very interesting.
Yeah, for me, I have studied about the ocean ecology, and ocean ecology. They said we have ecological change by the climate change, but in that time I have a question. So how we can solve this problem? So that's I want to get the answers about this climate change or future energy and environmental areas. So that's why I selected this bioenergy technology, and then once I started to the working in this bioenergy area, and yeah, but synthetic biology is a very fantastic technology to overcoming the current chemical technology. So yeah, that makes a very interesting for me.
Great, thank you both. Super fascinating how there's different paths to sort of get to close to the same field. Very interesting for folks out there. One question for Yuzhong, and maybe both of you, because I think this is a little bit of a tangential kind of subject, but totally good question. So this person says, California generates 56 million tons of biomass every year for forest thinning, agricultural, and municipal. Much of this is burned, sending CO2 into the atmosphere. How far away are we from being able to convert these sources into sugars that could then be used for biofuels and byproducts? So the biomass that's burned for forest thinning and agriculture, those sort of things, can we eventually take those to be used as biofuels? Because I know we're talking about other types of plant material, right?
Yeah, absolutely. So so far we have been working on mostly softwood deconstruction, but we're absolutely expanding the variety of wood and biomass that we can work with. How far away? Well, we're on our way, but I can definitely envision that happening in the near future.
Yeah, so in addition to Yuzhong's answer, so that's why we are trying to use the sugarcane or other waste material, because actually it is very hard to reach up the chemical industry. I mean, the productivity, bioproduction productivity to the chemical production industry. So, but we can decrease the cost or operation cost by using the waste material. But they are using the very expensive material, petroleum, but we are using the waste material, so we can decrease the cost from here. So maybe we can find a way.
Great, thank you so much for both of those answers. That was great. So one more question: are there efforts right now to separate hydrogen from water using bioenergy for use as a fuel? I don't know. There, okay, go for it, Changman.
Yeah, so I have been working in the two projects in ABPDU. One project is we are trying to produce a biohydrogen from the cellulosic or the cellulosic lignin or woody biomass to produce hydrogen. There is one strategy to produce a hydrogen, The other strategy is we can use the electricity and we can put the electricity into the bacteria, and some remaining energy should be converted to hydrogen. So we want to collect this hydrogen as a fuel or energy sources.
Great. Yuzhong, do you have anything to add there? Nope.
Not really.
Not really. Okay, so with that, I am going to thank everyone for being here. Again, our two speakers have been wonderful. I don't know, Jocelyn, do you have anything more to say? Just encouraging word to our speakers and our audience for being so engaging and active.
No, just thank you so much for joining us. It was such an engaging presentation. And if you'd like to stay up to date on any research coming out of our institutions, you can visit us at Science at Cal at science@cal.berkeley.edu and Berkeley Lab at lbl.gov. And I just wanted to remind folks about our presentation next month, which will be focusing on negative emissions technology.
Yes, thank you so much for joining us. Thank you so much to our speakers, and we will see you back next month. Thanks again, everyone.